Yes, sir.
Okay,
Is it glad you can share your screen now?
Thank you. Happy. Okay, now let's get into the structure of the bacteria, the structure, and the remaining parts of today's lectures. I give a more detailed introduction to the structure of bacteria as well as archaea, and then discuss some aspects of their metabolism. The first problem is the size of the bacteria. Generally, the bacteria is much smaller than the cells, not the eukaryotic cells. And the average size is 0.2 to 2 micrometer in diameter, and the length is 2 to 8 micrometers. So these are the general, many bacteria fell into this range. And in terms of shapes, there are not much variation in their shapes. That is, they are spherical, it's called coccus. If they were rod-shaped, it's called the bacillus. And if it's spiral, we call it's a spirochete or spirillum. And if two coccus are linked together, sometimes it's called diplococcus. And if they form a chain, it's called a streptococcus. If they form a grape-like cluster, it's called a staphylococcus. If they are short rod, it's called coccobacilli. So these are the vocabulary for describing the shapes of bacteria. First, let's talk about the size. Smaller size has its advantages. That is, things can diffuse into the cell and out of the cell very quickly. And also its surface to volume ratio is higher. So it can maintain a higher metabolism. And also it can have a bigger population, that is in a space, in a fixed space. Smaller size can support a bigger population of bacteria. So that the evolution of this population is faster. So that this is why bacteria generally remain small. And eukaryotic cells, the diameter is from about 20 micrometer up to about 200 micrometers. So in this range, that is the size can be thousandfold for eukaryotic cells than for small prokaryotic cells. So there's a much difference. And but since eukaryotic cells has many different kinds of adaptations, so both have its advantages and disadvantages. There are some large bacteria, such as one found in the fish, the symbiotic fish gut, fish intestine. So it's a spindle-shaped. So its length can be up to 700 micrometer, 0.7 micrometers. Diameter can be about 20 micrometers. So it's bigger than eukaryotic cells. And another, it's called a Epulopiscium. Epulopiscium. You don't need to remember it. It's Epulopiscium. That is, it's a fish gut bacteria. Another one is called Thiomargaritus. Thio means sulfur. Margaritus means pearl. Sulfur pearl. So it's a sea-side, that is shallow sea, pearl-like bacteria. And it has many sulfur granules in the cell body. So it's called a sulfur pearl bacteria. its diameter can be about 800 micrometers. So this size is bigger than the eukaryotic cells. So these are the exceptions. And besides these huge bacteria, most of the bacteria fall into a smaller range. That is, diameter is up to 2 microliters, length up to 8 micrometers. And but can we have smaller ones, the nanobacteria, or called the nanobu, N-A-N-O-B-E, nanobes, means that we have microbes, and then nano means 1,000 times smaller than this. So some scientists propose that there must be smaller bacteria, that is diameter about 0.1 microliters. However, if you consider that diameter is only 0.1 microliter, then the volume 1,000 times smaller than the average size diameter. So can it hold enough chromosome or enough ribosome for its metabolism? So it seems not very much likely. And so we have, up to now, have known that bacteria can be as small as 0.2 micrometer in diameter. So current thinking is that scientists put the small size bacteria, nanos, with diameter of 0.1 microliter. However, there are smaller bacteria which has a diameter of 0.2 or higher. That means that this bacteria must have some help from its host, because its metabolic ability is severely restricted. So they cannot use this. They cannot have many ribosomes. They cannot have many proteins. So their metabolism must be dependent upon their host. That is, wherever they parasitize, they use the host machinery to help them to reproduce. So these are the basic thinkings about the size of bacteria. Okay, now we can let me show you the various color-coded micrograph of bacteria. So on the left side is the coccus. It's Coccobacillus. This is Bacillus. This is the spiroform, spirochete or spirillum. Okay, so these are some very peculiar shapes of the bacteria. These are star-shaped. Star-shaped is called stella. Stella is star, and this is also star-shaped. And this is a brick-shaped. Rectangular shaped. Okay, so these are the some more strange shaped bacteria. Some are pleomorphic. Pleo means more than one. Pleomorphic is changing shape. That is, it's multi-shaped. That is variable shaped. It's called pleomorphic. So if the surface of the bacteria is not very tight or strong, then the bacteria can change its shape. So we can observe many different shapes of this bacteria. So it will be described as pleomorphic bacteria. Okay, these are the arrangement of coccus. Okay, so let me first introduce the general shape of the bacteria. The generally prokaryotic cell inside is simpler than eukaryotic cells. So you have learned that the eukaryotic cells has many organelles. And so the origin of those organelles, some can be explained by the endosymbiotic theory. Endosymbiotic means symbiosis for some bacteria inside a larger microorganisms. That is a larger cell which hosts many smaller microorganisms. It's called endosymbiosis. So this endosymbiotic theory can explain the origin of mitochondria as well as chloroplast. So these two large organelles of eukaryotic cells are the endosymbiosis of smaller microorganisms. Then other organelles, such as endoplasmic reticulum or Golgi apparatus, there's yet no, its origin has not been explained or has not been proposed in terms of theory. For the prokaryotic cells, the inside is generally simple. That means that this is a schematic. Schematic diagram means that we draw a picture to show its major points. Then the right side is the electromicrograph of bacteria. So that means that it's indeed much simpler. And on the left side, we have seen some noodle-like things. This is called a nucleoid. Nucleoid. Oid means like, similar to. So this is similar to a nucleus, the nucleus of the eukaryotic cells. But there is no nuclear envelope. So this area is generally called a nucleoid. And this nucleoid is thought to have DNA, and this is shown. This thread is indeed the DNA. But however, this DNA is folded many times. Because if we measure the amount of DNA in one bacteria and then calculate its total length, so this total length can be up to 1 centimeter long. And this 1 centimeter will be folded into this size. That is, this size is about 2 micrometers, right? So this is the folding is more than 1,000 times. So this will pose a great difficulty for us to understand, because how can these things be folded more than 1,000 times and still the information is easily taken and easily transcribed and translated. So this is quite an interesting phenomena. We know that bacterial cells respond to the outside stimulus very fast. That is, it responded to the presence of toxins, to the presence of nutrients, to the presence of osmotic shock. All these extremely fast, so they can turn on their genes and turn off their genes in order to cope with the environment. So this easy retrieval of the information, the genetic information, for its use is very interesting for us. However, scientists have not the tools to probe into this information retrieval problem for the microorganisms. So this is called the nucleoid. This in the electromicrograph, we can see this light-colored region. We call it the nucleoid. Okay, and then besides these nucleoids, we have some tiny particles. So these tiny particles are the ribosomes. That is the larger aggregate of proteins, which are used for the translation of proteins from messenger RNA. And so for growing bacteria, it has maybe thousands of ribosomes. For starving bacteria, we have only a few ribosomes. So these are the important cytoplasmic machinery for the bacteria. Besides these two ubiquitous things, that is nucleoid and the ribosome, some bacteria have some inclusions. Inclusion means some structures inside, and later on I discuss those inclusions, those bodies. So like this one or like this one, okay, and we'll go into more details about this. And the difference between the eukaryotic and prokaryotic cells One major part is the periphery, that is the envelopes of the bacteria. It is much more complex, generally much more complex than eukaryotic cells, because eukaryotic cells can cooperate. That is, they can cooperate as a population to build something to cope with the outside, such as if we human beings have a skin, and the skin has many layers to cope with the outside influence. But the one single bacteria is one organism, so they need to have a elaborate, that is very complicated outside structures to cope with the environment. And so I will discuss those things. First, let's go into the plasma membrane. And the plasma membrane, plasma means the cell, the cell basic material is in the cell. It's called the plasma. This plasma, the membrane enclosing it, it's called the plasma membrane. Okay, this is a schematic diagram. It shows three kinds of things. So these are very important because instead of one single plasma membrane, we can see that it is one inside and one in the middle. It's called peptidoglycan. Okay, then on the outside, so that some bacteria has two plasma membranes, two membranes. So this is a very strange, and which we have not figured out how and why. It has two membranes and one. This is wall, cell wall. So under the electron micrograph, we can see clearly this is a marvelous picture of the cell periphery. That is, these double real-like things are the plasma membrane, the inner plasma membrane. And so you can see two dark lines and one light lines. Okay, and this one dark line is called the peptidoglycan, which I will explain later. And this one outer membrane, you can see that it's not the same as the inner membrane. Inner membrane has two well demarcated, that is well delineated black lines. One, and this is fuzzier black lines. Okay, let me first introduce the structure of this inner plasma membrane. And you have learned in the introductory biology that this is called the union membrane, lipid bilayer. This is very important because it is this lipid bilayer is two layers of molecules, and they are facing. One is facing outside, and one is facing inside. And this is called the phospholipid. That's called phospholipid means that it has a phosphate on the glycerol, and then two fatty acids, which also link to the glycerol. So glycerol is the hinge. It's a hinge. Glycerol has three OH hydroxyl groups. One OH is linked to phosphate, and the other two are linked to fatty acid. This fatty acid is called hydrophobic region because it is hating the water. It means that because of the hydrophobic interactions, they like to cluster together. Okay, so this became the hydrophobic region. And outside, because of the phosphate, so it likes the water molecule. It's called the hydrophilic region. So this region is because it's phosphate, so it attracts calcium, magnesium. There's a positive ions as well as water, because water has a hydrophilic region and hydrophobic region. So the hydrophobic, the positive region, the negative region, and phosphate because it's negative, and then the positive region will orient towards this phosphate. So these are the theoretical structure. And this serves the barrier between the outside and the inside. So this is very important structure. And inside the plasma membrane, there are many proteins. And so the theory